I just counted, look at my presentation, a little bit over 60 slides to go through. So perhaps some of them I will just skip or go faster because there are some important messages, at least I want to get through. So okay, so you see how powerful it is to use this theoretical framework based on simplifying assumptions that we have a sphere, homogeneous sphere, and we develop this theoretical framework for the behavior of individual single particle properties as a function of particle physical and chemical properties of particle, and then apply this simplified, idealized theoretical framework to interpret our results that actually apply to actual non-spherical particles that have irregular shapes, that have internal variations in refractive index and so on, and we can still gain quite a good idea about how to interpret the features, for example, both magnitudes and spectral features of actual real particles. And one of these features is here in scattering, is this kind of effect in absorption bands, and I already indicated it a moment ago before the break, and it does relate also to the so-called anomalous dispersion of the refractive index within absorption band. So what is anomalous dispersion versus sort of normal or regular dispersion? If you have a substance and you look at the real part of refractive index of substance as a function of light wavelength, it's going typically, the refractive index is going typically to increase with decreasing wavelength. Why is that? Because if you send photons of higher and higher energies on a substance, the response of these dipoles will be stronger for higher energy photons, shorter wavelength photons. You will generate a higher dipole moment. That's where normal dispersion of most substances come from. So if you move towards short, visible, and UV, you're actually generating higher dipole moment and more higher refractive and higher polarizability and higher refractive index. But this general pattern can be perturbed if you're within a strong absorption band, something like this, if this is a wavelength or frequency and you have a strong absorption band. So real part of refractive index, this would be a pattern outside the absorption band here and there that would sort of, this is the wavelength decreasing to the left. And this would be a normal dispersion, and you have this oscillation, strong oscillation of a real part of refractive index. It's referred to as anomalous dispersion. Another example, experimental example of efficiency factors. This time we are combining on these two plots. Two graphs are for two different species, Platymonas, which is about 3.4 micron in size, and Cyanobacteria synecocystis is about the mean diameter. This is a small d. Okay, I'm not 100% consistent in notation between the slides, so I apologize for this. In this case, this is the mean diameter, and 1.2 micron, 3.4 micron, and you can see Q sub C. Just let's look at solid lines, which are experimental estimates, Q sub B and Q sub A. Okay, so now we have. We also can estimate real part of refractive index for these particles from optical measurements, knowing optical measurement of spectrum of attenuation and size. And if we know absorption, the single unknown is real part of refractive index. You can do some sort of iteration and try to estimate the real part of refractive index. So here was the real part of refractive index estimation, about 1.07. For this smaller guy, 1.05. Alpha, which is pi d times nw over lambda, it's in this spectral range is about 20 to 35 for this smaller species, 7 to 13. That's alpha, that's relative size. And then we can calculate what's the range of rho values. 2 alpha times n minus 1 for this species within this visible spectral ranges between 3 and 5, here between 0.5, 1.5. Okay, so let's recall our behavior of this theoretical curve for attenuation or scattering. Or we are in ascend for these small values of rho. We are on this ascending slope. So you can see the spectral shape is like the shorter the wavelength, the higher the attenuation or the higher the scattering. These values 3 to 5, we are actually reaching this first maximum of Q sub B or Q sub C versus rho. And these numbers are now high because the first maximum, they are pretty high, over 2. And okay, so we are within that region. So another very nice example, how these realistic particles, how measurements on realistic particles, non-spherical, heterogeneous, how we can interpret it and understand where we are in terms of physical properties of the particle. Okay, that slide is, I already wrote a definition for how you build beam transmissometer, how you measure it to get these Q factors, QC factors. Of course, beam transmissometer should just collect directly transmitted light. If it's an ideal beam transmissometer, we unfortunately always collect some forward scattered light at very small angles. So in the lab, we could use geometry like this. We have a detector. Our sample is as far as possible from the detector, and we have some apertures. So we only want to collect the directly transmitted light. There are different ways of doing this in situ instruments as well. You will work with one of them tomorrow. Okay, so I've been talking about absorption, total scattering, attenuation, efficiencies, and cross-sections. Okay, I just efficiencies times geometric projected area also. Okay, so these are important concepts. But this is only half of the story of what we want to know about single particle properties for different kinds of particles. Another thing is that these efficiency factors tell us nothing about the angular distribution of scattering. Okay, and I put some emphasis in my description on experiments. This is important because it's so hard to model accurately scattering and absorption of realistic particles, or very many different kinds of particles. So it is important to develop our understanding and knowledge and quantitative data based on well-designed and as accurate as possible experiments for these realistic particles. And we would also like to use the similar approach for characterization of the angular dependence of scattering. But here we have even less quantitative data, experimental data for different types of particles in terms of how the volume scattering function or phase function, this is a phase function, so volume scattering function divided by the total scattering coefficient. That's even more difficult in terms of experiments, just we don't have instruments readily available. There are a few, and you will learn a lot more about this kind of work on Friday from Mike Twardowski. He's going to talk the whole morning, 2 lectures on ocean light scattering, probably quite a lot about measurements of volume scattering function, phase functions. So I will show you some results, again, from meteory that gives us some idea, some guidance about what to expect, how the volume scattering function or the shape of volume scattering function changes in response to varying properties of particles. So here we have example of alpha. So one relative size is 12. And we have 3 results for 3 calculations for refractive index, which is just real part of refractive index, non-absorbing particles, 1.035. So not very small difference between the particle refractive index and the medium. There is another particle, which is the dashed line here, has a little bit of absorption. That's the imaginary part of refractive index. And this calculation includes some small degree of polydispersity. Ln means some log normal distribution around this value of alpha. So you can see now how the shape of scattering function varies as a function of angle for a particle with this relative size. Okay, alpha 12, it's quite large. We had example here. Well, this guy, 1 micron in size, had 7 to 13. So it's a particle larger than light wavelength with alpha 12. So you can see highly forward, this is log scale. So forward peak, this is the first diffraction lobe. We talk about it. This decay, or how fast it goes down, can be used for sizing particles using this diffraction, inversion of diffraction pattern. And you can see minima, maxima associated with this constructive, destructive interference. And some effect of absorption you can see in these dashed lines. there's a little bit of reduction of the magnitude of scattering, more so in the backward directions than in the forward directions. And you can see that small degree of polydispersity immediately smooths out the pattern associated with the interference pattern. So if we have particles of different sizes, the location of this minima and maxima shifts here to the right and to the left from different sizes, and that causes the smoothing effect. And these are the examples of computations of phase functions. On the left-hand side, we fix the refractive index and we change alpha. So we change size with respect to wavelength. So alpha 4, 12, 60, 300. This is the real part of refractive index. So okay, so let's take a look. If we have a small particle, alpha 4, it looks like this. So what happens is this is less forward peak. The magnitude are lower in the forward direction, well, relatively lower compared to the backward direction. So the ratio of backward scattering to total scattering or forward scattering is higher for the small size guys. If we increase the size, the shape of the volume scattering function or phase functions becomes highly forward peak. So that also indicates us the ability to size particles based on this decrease here of the function. It decreases very fast for very large particle and decreases slowly for small particles. Now, if we fix size at 100, by the way, we're using small degree of polydispersity to get rid of this interference pattern, the complex features. Now we get here 1.02, 1.03, 1.05, 1.075. Yeah, so this is the range of relatively low relative refractive indices, probably within the range that you can encounter within living biological cells. And you can see again, well, now you can see very interesting thing. In the forward directions, near forward directions, where diffraction is important, you don't see effect of refractive index on the shape of phase function. So combination of this guy and this guy provides a basis for sizing particles based on how fast this function decreases with wavelength for very small angles. So you can see no effect in the diffraction domain of refractive index. But particles are relatively large. It has to be relatively large. Huge effect here for refractive index. So you can see high refractive index produces significantly more scattering at large angles and backscattering angles compared to low refractive index. These are not measurements, these are calculations for particles that were assumed to be spheres, but may represent maybe close to something like bacteria, flagellates, ciliates, and this is chlorophyte Dunaliella. It's probably around 10 micron in size. So you can see for bacteria the function is, of course, it drops only maybe 3-4 orders of magnitude. With increasing angle, this drops by 7 orders of magnitude. Backscattering ratio. That's another parameter that is useful to look at. We just talked, Songping, during the break, that in some papers this backscattering ratio is basically the ratio of backscattering coefficient to total scattering coefficient, or for single particles, backscattering efficiency over total scattering efficiency. And in some papers, the term backscattering efficiency is used to indicate backscattering ratio. So there may be some confusion. It's better, I would say it's better to avoid naming this quantity efficiency, because efficiency is really efficiency. It's the power of backscattered power divided by the power incident on the projected area of particle. Okay, so again, here we have alpha, just alpha, so size of particle, and different particles are represented by different refractive index. So from 1 through 4, just real part of refractive index, non-absorbing particles, and here the same real part of refractive index, but there is absorption added. There's one example for monodisperse system, but all of these are for some degree of polydispersity. And what you can see here is that in the limit of alpha, very small alpha, the backscattering ratio approaches the value of 0.5. So we've already seen this today, this morning, the diagram, polar diagram for angular scattering of a very small particle or molecule, Rayleigh type of scattering for something that is smaller than 10 times wavelength would scatter as much light in all forward direction as would scatter in all backward direction. That's why this curve goes to 0.5 here. This is a log scale. You can see as we increase alpha, the backscattering ratio drops dramatically. In this range of alpha, there is not much dependence on refractive index, but it starts to play a significant role for alpha greater than 10 or so. So for things larger than about 1 micron or so, maybe less than 1 micron, it depends on wavelength, it depends on other things as well. But you can see very large dependence. So this is high refractive index, 1 is very low refractive index, or if you add absorption, things go down. Yes? So absorption will reduce the backscattering ratio even further. And would be great to have instruments and, add to our experimental designs in a more routine fashion the measurements of volume scattering function on different types of samples and particles, so we can characterize this. There are some papers in literature that provide this information. I'm not going to go over it, and I think Mike will talk about it more. So basically, you would need some sort of instrument that is measuring, sending a beam of light, this interrogated volume, and is measuring scattering at this angle. And you may have a bunch of detectors around, or maybe rotating arm, and all kinds of designs are possible. Let me now review briefly a topic of interspecies variability in optical properties. So we've seen this theoretical framework, how changing size refractive index affects the single particle optical properties. And some years ago, actually 20 years ago, I was here in Villefranche, and I worked with Annick and André Morel on putting together data from very many lab experiments that were done with different cultures in which we measured both absorption and beam attenuation. I think many people contributed, by the way, to our knowledge about optical properties of phytoplankton and other particles. So some of people are here in the room. Colin did a lot of work on this as well. Some of the work that was done those years here is characteristic in a sense that the measurements were done on spectrum of absorption, spectrum of beam attenuation, so you can get spectrum of scattering on the same culture, and size distribution counting, and also pigments oftentimes, and things like this. So we decided to use these experiments from the past and put together the database for these different groups of phytoplankton, sort of organize it and see whether we can use it in some way to learn a little bit about, potential roles of different kind of species in optics and things like that. So we have things ranging from viruses and heterotrophic bacteria in submicron range, Prochlorococcus, then cyanobacteria here around 1 micron, and several species, larger species from about 3 microns to about 30 microns. So I'd like to show you how the data look like for several quantities, optical properties. So here we have chlorophyll-specific absorption spectra for all these cultures. I don't name which one is which, but just want to make a point that there's a significant variability in amount of absorption per unit chlorophyll between different species. And you can see, of course, not only magnitude, but shapes are different because of different accessory pigments and things like this. For example, this is phycocyanin peak associated with this phycocyanin ridge. These things. And okay, so this is normalization of absorption to chlorophyll. If we normalize absorption measurements, absorption coefficients to the number concentration of particles, you get the cross section. This is this meter squared per particle we discussed. So this is, in other words, the product of the efficiency factors times the geometric projected area. Now you have log scale here, so you have pretty significant variation, interspecies variation in cross section. Or it is driven by what? Mostly by size. Yes, remember rho prime is the AS or N prime times the size. Well, N prime for sure varies to some extent between the species, but the size varies quite a lot because bacteria here, heterotrophic bacteria, are 0.5 micron in size, and this species is 30 micron in size. So this rho prime parameter really is much larger for this species. So size is a major driver here. And if we normalize these spectra, these are the same, if we normalize either this spectra or this spectra to unity, somewhere here around 400, you can see variations in spectral shapes. So all these guys are photosynthetic phytoplankton. There's one curve that's quite different, looks like this. That's for heterotrophic bacteria. So it's weak cross-section, low magnitude, and it's very distinct shape, quite different from pigmented phytoplankton. I will get back to this, but it has interesting peak here around 410, which is associated with cytochromes, we believe. The same story for scattering, chlorophyll-specific scattering here. Big variation between species and huge variation in terms of cross-sections. Well, these are viruses. Viruses were not included in the previous. We just assumed non-absorbing, that the absorption was so low we didn't even bother with this, but these are viruses. Well, this would be very close to lambda minus 4, because these viruses were about 60 nanometers, not quite yet, but in fact, if I recall, 3.8 something. So almost Rayleigh type of scattering of these small particles suspended in water. Bacteria, cyanobacteria, and other stuff. But you see a lot of orders of magnitude. Again, you can see here variations in spectral shapes, viruses, very steep, somewhere there, heterotrophic bacteria. All kinds of phytoplankton show this inverse sort of pattern to absorption, yes, in terms of scattering. Single scattering albedo. I don't know if it was mentioned when IOPs were discussed, something that is useful. It's called also probability of photon survival. It's a ratio of scattering to beam attenuation. So if there is an interaction between photon and a particle, two things may happen. It can be attenuated, it can be scattered or absorbed. So that basically, if it's absorbed, if it's scattered, the photon survives. If it's not, it doesn't. So the single scattering albedo is B over C, or QB over QC. For single particles, you can see variations in the single scattering albedo. It reminds us some kind of absorption spectrum, but the other way around. Yeah, so I mean, when there is very little absorption here, the photon survival probability of photon survival close to 1. This, I believe, was Prochlorococcus culture, which was highly pigmented. Well, this is probably one of the very unique data showing that in some phytoplankton, the single scattering albedo can be lower than 0.5, even 0.4. If the cell is highly pigmented, it has huge absorption. So probability of photon survival fairly low. Single scattering albedo is also a parameter that's often used by people in radiative transfer theory, and sometimes the radiative transfer equation is written with this parameter involved. I think atmospheric people like to use it a lot. Okay, so for all these groups, we don't have measurements of phase functions, but we could use the size, estimate of refractive index, estimate of imaginary refractive index, and assume that these are spheres. So let's assume that the horse is a sphere and compute the scattering phase functions. They would look like this if they were spheres. In reality, this is wrong. This is not actual phase functions of these real particles. We know that by assuming that the particle is homogeneous sphere, we underestimate, typically we underestimate backscattering in relation to forward scattering. So we underestimate the backscattering ratio, for example, by ignoring the fact that the particles have some structure, internal structure. There's several papers documenting this. There are theoretical papers showing one-layer spheres with one layer, or two layers, things like that, and some other evidence as well. Okay, because we don't have measurements, or at least there are few measurements, not at least in this database, the measurements of volume scattering function, the estimation of backscattering properties, something like backscattering cross-sections, or was also prone or subject to significant uncertainty, because we would have to assume the me solutions for the scattering function. So I'm not going to talk much about this. Okay, so that was something about the interspecies variability. You see a range. A few slides, maybe 3-4 slides, about the fact that things are even more complex, because these are living guys, and they change their properties in response to environmental conditions or to growth conditions. If you grow them under low light versus high light, you will experience, or you will see, changes in optical properties, changes in size, changes in refractive index, or other factors, environmental factors, such as temperature and availability of nutrients, can also cause differences. So this is an example of an experiment in which you have Q sub A, Q sub B, chlorophyll-specific absorption, chlorophyll-specific scattering spectra, and carbon-specific absorption and carbon-specific scattering spectra for one species, Synecocystis. This is a cyanobacterium with phycocyanin pigment. And these numbers, each curve is accompanied by a number here, 20, 40, 80, and up to 1450. This is par. So you already know what par is. Par in micro-Einsteins per meter square per second. So you see this is a very low value of par, very dim light, 20 micro-Einsteins per meter square per second. 1450, this would be full sun, more or less. That's a very bright light. So these cultures were grown under different conditions. They were acclimated to different light conditions. They were growing exponentially, so very happy, healthy, plenty of nutrients, good temperature, optimal temperature. So the only limiting factor could be light. And you can see how different optical properties, depending how you normalize them, vary between different environments. So one thing, for example, is here that the efficiencies are very high for low light adapted cells here compared to high light adapted cells. Makes sense. They synthesize pigments, if they're more pigments, if they're under low light. What else? For example, in terms of carbon specific coefficients, you can see that we reduce the variability quite significantly compared to chlorophyll normalization. So that actually was one of the early results that led us to believe that scattering can sort of be a proxy for amount of carbon in biological particles. Another example of intraspecies variability, where now the species is a diatom, small diatom, about 4 micron in size, was grown under optimal conditions, then temperature limitation, nitrogen limitation, light limitation. You can plot things like cross-section for absorption, or chlorophyll-specific absorption, cross-section for scattering, and carbon-specific scattering versus growth rate, which is kind of interesting to try to look into potential relationship between growth rate associated with different growth conditions and optical properties, whether optics carries any information on growth of cells. And you can see this is a light limitation behavior for absorption. So under slow light limited cells are growing slowly, but they exhibit high cross section for absorption because they synthesize a lot of pigment. And and because of that, actually, when you normalize to chlorophyll, this point is below the other conditions. And other conditions the nitrogen limitation and temperature limitations are almost very similar pattern. Okay, so the acclimation to these different conditions cause variations for any species of phytoplankton, but phytoplankton grow in most environments under pretty significant diel cycle in incident irradiance. Yes, maybe with the exception of very high latitudes during summer or things like that, but for the most part, they would experience darkness during the night and daylight during the day. So it is of interest to see whether phytoplankton changes properties, optical properties over a day-night cycle. And there was some work, early work, that indicated that actually such variations in diel variations in optical properties were observed in situ. For example, in beam attenuation coefficient, it was hypothesized that it may be a measure of just of variations in concentration of cells over a day-night cycle when they divide, let's say, near the end of the day in some sort of synchronous fashion, things like this. But it is interesting to look in a greater detail in potential sources of diel variability in optical properties. And this is an example of work where you see measurements taken over 3 days and 3 nights in the lab, also on this species, where you can see every 3 hours or so the measurements were done of the size distribution. So this is from size, this is night, this is the sunrise, and this was in March, so about the same length of the night and day, and the culture was exposed to natural light rather than some artificial light produced by lamps. But you can see decrease in size during the night, increase in size the first part of the day, and then decrease, increase. Very nice consistent pattern over 3 days and nights. Then you could estimate real part of refractive index of these cells from scattering measurements, from size, through some iterative process, and you can see how refractive indexes changes nicely. But there is some phase lag between size and refractive index peaks later, just near the end of the day. And okay, so we know now from previous plots that scattering cross-section is driven largely by this rho parameter, which involves size and refractive index. So you can see that scattering cross-section of this species varied over day-night cycle dramatically, twofold. That's huge. This is the same species. Yes, at the sunrise, it has a very low value, and then in a matter of 3-4 hours, it increases its capacity to scatter light by a factor of 2. Nothing to do with the increase in particle concentration in our suspension. It's just the properties of single particles change. So what happens here? It increases the cells, increase in size. This goes up. Increase in refractive index also drives it up. The cells were dividing near the end of the day. And then the size was decreasing, and refractive index is decreasing during, it's increasing during the day because cells photosynthesize, and if you wish, they get denser. They get more solid material per unit volume of cell. And during the night, there is respiration, so they get less dense and lower refractive index. So these things are important to take into account when you take measurements in the ocean and want to interpret the optical measurements in terms of variations in your bulk. Scattering coefficient can vary due to changes in concentration of particles, but also can change due to changes in single particle properties or both. Yes, so this is the indication that single particle properties may play a role. I'm skipping a few slides here. Yes?
And it has been observed, I'm not sure if it's any way to observe these, but they're quite fast, these changes. It's possible that you have a mixed layer that is mixing not very fast. These optical properties are changing while the phytoplankton is drifting inside.
Yeah.
Is that possible?
Could you just repeat the question?
Yeah, the question is about the observation of that these dial the observations of dial changes in the ocean are affected also by mixing on time scales comparable with what we see here, possibly the mixing within the, so they are exposed really, the cells are being exposed to different levels of light as they are mixed deeper within the water column and coming back toward the surface. So there's another factor in real world that plays a role that's not, of course, taken into account here, where what you see here is just the effect of the variation in the daily illumination and how these cells respond to daily illumination. So,
But it's possible.
yeah. So, I mean, the light environment to which cells in a real world are being exposed to is not exactly what is here, because this would just possibly simulate something that happens near the surface over the entire day-night cycle without cells being mixed into deeper layers. So it may not be as strong as shown here. Okay, let's take a quick look at optical properties in another group of organisms, heterotrophic bacteria. So these are small particles, less than 1 micron in size. And okay, so normally, historically, the role of these particles have been neglected for absorption, but it was some 40, 30 years ago. It was not clear whether they might be important to scattering. So some work has been done on heterotrophic bacteria, try to estimate these cross-sections and efficiency factors. And you see example data here, where there's absorption cross-section and attenuation cross-section, where basically these numbers are quite small because this is multiplied by 10 to 15, so it's in fact 10 to minus 15. But okay, you can see this. This is a typical spectrum of cross-section for non-pigmented heterotrophic bacteria. This is what we think they are typically. If you isolate heterotrophic bacteria in the lab on some plates, they don't really show color when they grow as colonies. But it turns out that sometimes when you isolate heterotrophic bacteria from seawater sample, you see patches of colonies that have sort of orange color. And it is known that heterotrophic bacteria may contain carotenoids, pigments, and you can see measurements in some cultures like this, and they have significantly higher cross-sections. But to this point, I don't know whether, and I don't know if anybody knows, when I talk to microbiologists, they could never ascertain whether how many pigmented heterotrophic bacteria with the carotenoids versus non-pigmented are in seawater. So I think this is one topic, perhaps for one of you at some point in time. That could be interesting to look at, because if indeed pigmented heterotrophic bacteria are abundant in seawater, then they might have some measurable contribution. But these are normalized spectra of heterotrophic bacteria. You can see these are the typical spectrum of non-pigmented heterotrophic bacterium at this peak here, around 410 due to cytochrome, and these are the spectra with carotenoid-rich heterotrophic bacteria, and this is nothing surprising. Bacteria have attenuation cross-section that increase towards shorter wavelengths because they are small. Another aspect of particle optics that has not been well investigated, I believe, is, of course, the particles, biological particles, continuously interact with one another in some way, in natural environment. One of the interactions are prey-predator interactions, and we don't know exactly. There's not much work, at least I don't know, of work that really would somehow. There's some papers that would address it in a quantitative fashion, potential effects of what happens to a sample of water if you have some significant interactions of this kind of interactions. So this is an example again of lab experiment in which two groups of organisms have been used, cyanobacteria as a prey and ciliates as predators. So this is 1 micron cell. These are the microzooplankton particles, about 30 micron in size. They were eating these guys, and you start the experiment with a size distribution that looks like this, So these are cyanobacteria present in your suspension. And then you see after 2 weeks and 4 weeks, you see how they disappear from the suspension. So basically, they're all gone, right? They were all eaten. And you look how absorption and scattering properties of that suspension changes. And you also see dramatic difference in absorption, which is at the beginning dominated by cyanobacteria. And at the end, it looks more detrital, right? Non-algal particles. So the grazing is basically process of grazing. It tends to change the shape of absorption spectrum in this way. And it also, in this case, actually clears the suspension quite significantly, because at the end of the experiment, the whole suspension was less turbid than it was at the beginning. Okay, these are some other things related to scattering spectra, how they got flatter at the end of the experiment. So it means now larger proportion of larger particles is contributing to the spectrum at the end of the grazing experiment. Here is another example of variations or effects of interactions between biological particles on optical properties. And this experiment has been done on viral infection of marine bacteria. Actually, Julia is here, yeah? Julia has put a lot of effort to this experiment. But the idea was basically to look how the optical properties of suspensions are changed if your suspension consists of marine bacteria that are being infected by viruses. And what you see here on the right-hand side, this is time of the experiment. Now the time scales are much smaller than the other grazing experiment. These are hours, so this is the initial T0, 72 hours. And one of the important efforts in this experiment went into measuring the size distribution over a very broad range of sizes. This is diameter. You can see the effort was made to measure sizes as small as 50 nanometers or something like this. So we wanted to capture the population of viruses, and then other techniques. So this method covered the submicron particles, and there were 2 other methods we used for sizing, from less than 1 micron all the way to over 100 microns. So you could see what's going on. So initially you have this one. So these are bacteria that were the victims of this whole thing. And here are viruses. And you can see how viruses go up, and this peak of bacteria is decreasing. But you also see interesting stuff going on over here, because this is the optically important size range. So you can see what you generate through breakdown of bacterial cells. You also generate through some aggregation processes. You generate other particles that affect optics. And this is the reference with just bacteria without viruses. You see how stable and nice it is over time. So these effects are caused by production of particles due to viral infection of bacteria. And you can see that we started with beam attenuation coefficients like this and went down. It means that the suspension was cleared quite nicely through that process. Okay, so several slides about other types of particles. So at this point, I'm going to switch our focus on from biological particles on other types of particles, mineral particles that are everywhere in different proportions. They can get to oceanic environments through different ways, through deposition from the atmosphere, through in high latitude areas, through melting ice and things like this, or even very long-range atmospheric transport. Of course, rivers and things like that bring a lot of material into the ocean. Resuspension as well. So, okay, so another group of particles with huge diversity, many mineral species. I guess a few thousand, in fact, exist on Earth. And so not an easy task really to address, but we want to learn something at least about it. And we would like to isolate certain types of particles to learn about their properties. And that's the goal of some experiments that you can do in the lab. So this is an example of experiments with natural assemblages of particles that were dominated by minerals. It doesn't mean that they were entirely minerals. Some of them, they were like illite or kaolinite. These are some pure minerals, calcite, quartz. But these are the assemblages of particles which very small contribution of organic particles. You can test it. You can measure organic carbon in these particles. You can measure total mass concentration of particles suspended in water. You can measure organic carbon concentration, and by ratioing you can get an idea whether you have significant or insignificant effect of organic particles. So they come from different environments. One actually was collected here during a red rain event when the Saharan dust came over Villefranche, and you could see a beautiful red powder settling everywhere in Villefranche. And okay, so again, sort of a big mess, right? If you study the different types of samples and you measure the absorption spectra, you measure scattering spectra, and you measure, in this case, the most convenient way to normalize to something, would be because these are very polydispersed systems now, so to use the mass concentration of particles. So I think in remote sensing language, many people call it total suspended solids or something like TSM or something else. I like to call it mass concentration of suspended particulate matter, SPM. Total, to me, means that you collect everything, all particles, and I think we never collect all particles in this operational treatments, exercises. When you collect particles on the filter, I prefer to call it suspended particulate matter because you miss quite a bit of particles anyways through the filter. Anyway, so you measure absorption coefficient, you measure scattering coefficients, as I described before. You measure total mass of particles collected on the filter, the volume filtered, so you normalize and get this meter squared per gram. Variations is quite big between different samples. I want just to point for both, for scattering and absorption. But there is one interesting point I want to make. You can see two samples of, let's say, Saharan dust, SaH1, SaH2. And you can see how absorption of these two samples is different. This is the same powder, the same original sample. But these two measurements, one of these samples was subject to some change in size distribution, like this. So Saharan 1 sample contains relatively higher proportion of larger particles than Saharan 2 sample. But chemically, I mean, the particles were more or less the same. We can assume that imaginary part of refractive index sort of is probably the same across these sizes, but they are different in sizes. So, okay, so Saharan 1 has more larger sized particles. Saharan 1 sample has lower absorption per unit mass concentration of particles. This is something reminiscent of what we talk about package effect for phytoplankton, isn't it? The chlorophyll-specific absorption coefficient was lower for larger particles than smaller particles for phytoplankton. And we observe the same phenomenon here for mineral particles. So mineral particles seem to exhibit same sort of effect of package effect. So the mass-specific absorption of mineral particles also exhibits package effect here. You can see different colors of this for these samples if you collect them on glass fiber filters. Where is Saharan dust? Somewhere here. These particles, some of them reddish, brownish more than others. And this is an important observation because if you study different types of particles from different locations on Earth that are dominated by minerals, and you normalize them to total mass of particles, you get very large variation in mass-specific absorption. But if you normalize them to the amount of iron present in particles, you reduce this variation significantly. What it means that iron has a major absorbing compound in mineral-rich particles, and it absorbs primarily in this spectral region from about 500 something towards the blue and UV. So small addition of iron can have, actually the concentrations of iron in very many samples is very small, but it's enough to make a measurable and significant effect on absorption spectra. Okay, yet another group of particles that I wanted to talk about a little bit are submicron particles, colloids, nanoparticles, you can call them different ways. What kind of particles fall in this range? The smallest photosynthesizing particles from biological point of view. Bacteria, viruses, all kinds of organic detrital particles and tiny mineral particles will also fall in this range. There's not much data, unfortunately, about the number concentrations, size distribution in submicron range. It's very hard to measure with these techniques such as laser diffraction technique or Coulter principle. So electron microscopy has been used by Wells and Goldberg years ago to produce size distribution below 0.2 micron, or you can see in different environments have quite different size distribution. There's particle concentration, so how many particles per cubic meter versus size. And okay, that electron microscopy only is within the range from up to about 200 nanometers. But what about up to 1 micron? That's also tough to measure. These are data from work of Japanese scientists. What they use instrument that works the same principle as Coulter counter, and they publish data like this, down to 0.4 micron. It's very hard. Well, quite a few people who work with Coulter counter know that it's not easy to measure anything below 0.7 micron. So I myself, I'm not convinced whether these data are very good or not. But anyways, these are the data, very rare. You can rarely find any data like this. So if you use, so okay, we cannot, in situ, we cannot isolate the effect of colloids on optical measurement. This is a bulk measurement of absorption or attenuation or scattering. And if we count particles or size particles in the ocean, we also, if we don't have the ability to do it in submicron range, it's very hard to determine what is the role of colloidal particles in optical properties. But you can use this kind of data, assume that these particles are a horse is a sphere again, assume some realistic refractive indices, and for this kind of size distribution, you can compute what would be the scattering coefficient or backscattering coefficient produced by this population of submicron particles. You can compare it, for example, to pure water scattering or backscattering, and see how it looks like. So this was done in this plot, and you can see on the left-hand side scattering coefficient, on the right-hand backscattering coefficient. Well, there are many cases of these colloidal measurements, but here you can see average sort of situations based on all these measurements. So average small colloids below 200 nanometers, average large colloids from 400 nanometers up to 1 micron. So these are the data here. And by the way, small and large colloids is the solid line. So these are the large colloids, all colloids, pure water, and small colloids. So you can see in terms of total scattering, okay, in most environments, pure water does not contribute a lot to total scattering. The particles dominate, yes? So we would say that this is circumstantial evidence that small colloids are not important, but larger colloids may be important to total scattering. Now, in terms of backscattering, a little bit different story, because in many environments, as Zogby yesterday emphasized, in many environments, the pure water backscattering can be a dominant, or open ocean environments can be a dominant or very important contributor to total backscattering, especially at this wavelength here. And we can see that small colloids, okay, they are comparable here to water, so they may be, even small colloids can be important, larger colloids comparable to water. And you can see that these colloids are above, all of them, sum of these and this, so all submicron particles. So this is average based on all those plots of size distribution on previous slides. You're above water backscattering, and you're kind of close even in this region. So that would provide circumstantial evidence that colloids can be important to backscattering.
So, and we can suppose that these specters will change with time because these particles will distribute in the water, so the area concentration will change.
Yeah, so, okay, colloids are a very dynamic part of the particle assemblages. You can have spontaneous formation of colloids from macromolecules. You can have production of colloids from larger particles due to biological interactions, grazing, and all kinds of things. You can have dissolution, so it's a highly dynamic thing, but you always have them, and these are the most abundant particles in water. Okay. So always, regardless of, so if you take in 1 cubic centimeter of water, you may expect clear water in the middle of the ocean. You may expect, I don't know, at least the submicron range, 100 million particles, maybe even more. Easily, yeah. But you can see variations here, like these people measured. I mean, it's not the same. I mean, this is a log scale, so it can be more or less, so it's variable. It's always there, it's always most abundant range of, size range. Okay, so we don't have really a very solid knowledge about the contribution and role of differently sized particles. We can have some sort of indirect studies that suggest to us that this may be important or not, and certainly under many scenarios it can be important. And another example of this kind of exercise, but it's theoretical, is that you can assume size distribution in the ocean, all particles going from colloidal size ranges, 50 nanometers or so, all the way to hundreds microns, entire optically significant range. You can assume one wavelength for simplicity of discussion, one refractive index. You may assume no absorption, or you are in this range. Maybe for biological particles, no big deal if you assume that absorption is very weak. And you assume that the number of particles per unit volume decreases with increasing size with the power diameter to power -4. It oftentimes decreases -3 to -4.5 or something like this. There's the range of the slope, and it's variable. But okay, for the sake of simplicity of the model, you may assume different refractive indices and run these Mie calculations again and look at the contribution of different size fractions to total scattering and backscattering for this kind of population of homogeneous spheres. Now, this is not real marine particles, but homogeneous spheres. But still we learn something very interesting from this, because you can see here, this is a pi diagram, so this first triangle here represents contribution from 0 to 1 micron, from 1 micron to 2 microns, 2 to 3, 3 to 4, and so on. And this is for total scattering, this is for backscattering. So this is the contribution from less than 1 micron particles. Huge compared to this one. This may be whatever, less than 20%. Here you have over 75% contribution of submicron particles for this idealized assemblage of particles. And if we go to high index particles like 1.2, you still have a huge effect of submicron particles on the backscattering, and more effect for high index particles for total scattering. So this kind of work, suggests to us that it makes sense. It actually is important to dig in deeper, to design perhaps experiments that would be able to address the hypothesis with respect to the contribution of differently sized particles to scattering and backscattering, and learn more about the budget in terms of different particle size fractions. So that's another area for interesting work. Okay, so I want to show you something interesting in terms of how do we measure colloids these days. the data I showed was kind of difficult to get through electron microscopy. I want to show you some a few slides related to measurements. Okay, so maybe only once we run this. You can see these little dots dancing around. So this is Brownian motion of nanoparticles. So this is being measured with the system. In this case, it's not seawater, but there are 3 different polystyrene beads of different sizes, 50 nanometers, 240 nanometers, 800 nanometers, huge range of sizes. We cover most of the submicron range. And we know from our curves of efficiency factors or cross sections that there is a very steep curve for very small raw values, yes? So if you increase the size here by a little bit more than 10-fold, the scattering cross sections of this particle is like 6-7 orders of magnitude higher than scattering cross section of this guy. So these particles produce a very dim scattered field, and these particles produce incredibly bright scattered field compared to the small guys. In addition, I will run the movie one more time. Just notice some smaller images. This is actually not what you see. It's not the actual particle. It's the image of scattered light produced by the particle. The particles, the assemblage of particles is illuminated by the laser beam, and the signal is scattered signal is being collected at 90-degree scattering and projected on the CCD camera, and you get the movie. So well, you see how fast this small moves and how slowly larger moves. That's the very important aspect to this technique. So you can so by taking movies like this with the system, with this kind of a system, you can track during a movie each individual particle, its movement. You can compute its Brownian diffusion coefficient, and from Brownian diffusion coefficient you can determine its size. You can, if you have the calibrated system in terms of interrogated volume, you can also count these particles. You can determine the concentration, number concentration of particles and size distribution in this size range. So hopefully these technologies like this can advance, our understanding of the colloidal size range in the ocean in the near future. This instrument has been, by the way, commercialized, just commercialized, basically. And you can see here 300 video frames acquired during 10 seconds. So each of the clusters is one nanoparticle undergoing Brownian motion. So you see trajectory of the individual particle through time. And I have a privilege to display for the second time something about this miracle, year of miracles of Albert Einstein. If you remember, I brought it up when I talk about photoelectric effect. This was first of his papers in 1905. Second of his 4 papers was actually on the movement of small particles suspended in stationary liquids required by the molecular kinetic theory of heat. And Einstein talks here about this Brownian molecular motion associated with where does it come from? Molecules of the liquid under any given temperature. They move in chaotic random thermal motion, and they collide with small particles, push them around, and that's where the movement Brownian motion comes from. This is really a great, very elegant concept, because this is the equation that links the size of the nanoparticle in Brownian motion to the diffusion coefficient in Brownian motion. So this you really need to measure here. To get this thing, you need to measure the distance travel of the particle over a period of time, and you can get this diffusion coefficient. And you can see that the size of particle depends on the diffusion coefficient. So if we can measure this from these movies for each individual particle separately, all you need to know is the viscosity of the liquid in which your particles are suspended, and temperature of the liquid, which you can measure. Viscosity you can know. If we do it in seawater, at any given temperature, salinity, there are tables, you can get viscosity of seawater, right? We can measure temperature, and this is the Boltzmann constant. We've already seen it before. So what's great about this relationship, you can measure this, this stuff, and you compute the size for each individual particle. You can see it's independent on the type of particle. So you can have 50 nanometer gold particle and 50 nanometer virus, and it will diffuse the same way in a liquid with this temperature and this viscosity. Great. So you can size particles of different kinds just by measuring the Brownian diffusion coefficient. Okay, so we move on to the probably last component of seawater that I want to talk a little bit about is bubbles. Okay, so no surprise here. If you've been at sea or anywhere where you see breaking waves, you know that bubbles can dominate scattering in such situations. And here you can actually see the depth, 1 meter and 7.5 meters. And this is time. And the measurement, this is acoustic measurement when you measure concentration of bubbles. So the instrument is sitting below and sending the acoustic signal towards the surface, go up and down. And the color here indicates the concentration of bubbles. So you can see these events of bubble clouds being entrained into the water column, and then the water is almost devoid of bubbles, and then another entrainment, and so on and so forth. So this for people like we are here, for optics people, this is really a hard thing to work with. I mean, it's such an intermittent problem. I mean, they can dominate inherent optical properties, especially scattering, most importantly, not so absorption, but scattering here, and then, you know, 30 seconds later, they're not there. So this is the tough part of our work that actually hasn't been addressed in a very rigorous way yet. But we can sort of try to estimate, at least roughly, what to expect with regard to contribution of bubbles to scattering and backscattering coefficients for different void fractions. So by void fraction, I mean how much air is in water, right? The volume occupied by air or by air bubbles per unit volume of water. That's void fraction. And void fractions can be huge if you're measuring it here, just near the surface when you have a breaker. But if you're, you know, half a meter below or 20 centimeters below a breaker, or not necessarily during the event, but a few seconds after the event, these void fractions go down pretty quickly, but they are still significant. And you can see here the computation, and bubbles are relatively easy to model, easier than other particles, because we can assume sphere, we can do me calculations. Okay, there is a problem. Bubbles very quickly can get coated with organic substance, and we don't know exactly what to assume about that. You would have to assume a very tiny layer and some refractive index to this. So these computations, what you see here, are for uncoated bubbles, clean bubbles, but based on measurements of actual concentration and size distribution. So size distribution of bubbles was measured, concentrations, and me theory was run to predict scattering coefficients of a population of bubbles under different void fractions. You see, the void fractions here are less than 10 to minus 4. So it's 0.01% only air occupies within a given volume of water. So they're pretty small. So they are not within a breaking wave. So that's important to keep in mind. And you can see, for reference, you can see here pure seawater, total scattering, pure seawater, backscattering. For reference also, you have some prediction of scattering coefficient from sort of a model that predicts or estimates total scattering coefficient from chlorophyll concentration. So you would see here how that for different chlorophyll, this is low chlorophyll concentration, 0.1, 1, and 10. This is what would be the scattering coefficient associated with particles for these different chlorophyll concentrations, pure water scattering, and these are bubbles, right? At these void fractions, you can see at 10 to minus 4, and it is even high concentrations, the bubbles are even higher than particles. For backscattering, 10 to minus 5, 10 to minus 6, in this range you start to be comparable to pure water backscattering. So at very, very small void fractions, less than 10 to minus 4, bubbles may potentially dominate backscattering in the ocean. Okay, I have now 15 minutes and a little bit more philosophical. We'll talk briefly about traditional approaches, how we parameterize seawater composition in terms of particles. This is a traditional approach that, or lazy, easy, or whatever we like to call it. Songping called quite a bit about it when any IOPs would be pure water contribution particles and dissolved materials and particles. In the best case scenario in these approaches are divided into two components, phytoplankton, non-phytoplankton. But operational, this would be extractable portion of cells and non-extractable. I think Colin made a comment about how we do it with methanol extraction, things like that. But the bottom line is, okay, we assign this kind of categories, the names. We like to think about this as being phytoplankton and this non-algal particles and just a few components, right? Yeah, if you have a few components, so you have this kind of curves, typical curves for a 4-component model. Water, phytoplankton, sedum, and detritus. So water looks like this, total absorption looks like this, or phytoplankton would look like that. And these two guys, this is semi-log plot, so the spectra could look like this. Of course, the magnitudes vary, proportions vary, but you get a feeling here what are the kind of generic shapes of things. I think these plots were already shown. So total phytoplankton absorption, detrital absorption, and I think this is chlorophyll-specific phytoplankton. Maybe I will stop for a second here. Example on non-algal particle absorption from different environments. These are the data that were collected by Marcel and his collaborators around Europe. You can see generally this kind of almost featureless spectra in different environments for non-algal particles. Not always featureless. And you can study the slopes, some statistics of the slopes. It can carry some useful information, which is shown here. The similar analysis you can do for sedum, which is almost featureless as well, with different slopes, generally statistically different slopes than non-algal particles. Okay, so this is one approach. Another approach that has been around for several decades. Very powerful approach which has driven and motivated a lot of research. And actually we're sitting all today, I believe, just because of this kind of equation assumption that IOPs can be parameterized to relatively good, reasonably good accuracy by a single parameter in the ocean. There would not be ocean color science, any satellites, anything like this if 50 years ago a few people didn't kind of postulate this kind of relationship. So chlorophyll-based approach, very important. And there's one concept that I hope you are familiar with or heard about it, case 1, case 2, waters concept that has been introduced by André Morel and with some other contributions, where the waters where optical properties are driven largely or correlate well with chlorophyll concentration and everything, and if that correlation is kind of violated or is not as good, then this is everything else. So you may have waters affected by constituents that are not co-varying with chlorophyll. Some environments like near the river discharge or melting ice and things like that. So for case 1 waters, one would expect relatively good correlation with any optical or some optical properties and chlorophyll concentrations. And indeed, correlation coefficient is pretty high. If you collect a lot of data over a broad range of chlorophyll concentrations, but if you zoom in on portion of it and look at the variability, it can be pretty high. So a factor of 2 or 3 even, in this case 4-fold variation. For any given chlorophyll, you may find a significant variation in optical property. And this is similar variation, but for beam attenuation. So that may be even bigger than for absorption, for obvious reasons, yes. But the point I want to make that This was a very powerful observation, and it was published by Clark, Ewing, and Lorenzen in 1970 in Science. I believe that was Science paper. Though they didn't make many measurements from aircraft, not many, but enough to show the relationship between upwelling light and versus wavelength, and when they flew over waters with different chlorophyll concentrations, they've seen a distinct pattern in behavior of the spectra. So if you go from very low chlorophyll, you can decrease here in the blue, but not much change in the green, right? I think Zhongping mentioned that the original work at that time to develop first algorithms or relationship like this was based on, what, 37 data points or something like that, I don't know. You know, as time went on, this concept has been applied by researchers, and more and more data were collected, these band ratios data and chlorophyll concentrations. And at some point in time, in 90s, I think late 90s, we already had a few thousand data points to create this kind of algorithms that NASA called AEC4 algorithm that relates chlorophyll concentration with these band ratios of reflectance. So this has had a tremendous effect on our field, this kind of work in 70s. And it culminated with NASA decision to build coastal zone color scanner and run the experiment, the satellite mission on Nimbus 7 between 78 and 85. And this is a nice picture of CZCS. It was actually deployed on Nimbus satellite, which carried quite a few other instruments. And here you can see probably one of the first, if not the first, composite image satellite global distribution of phytoplankton from CZCS. CZCS didn't provide systematic data every day, everywhere, but over the 7 years of this proof of concept study, you could get this kind of data, which was a breakthrough. Well, later on, you know, more effort came, and CWIFS was being was developed by NASA and launched on Seastar spacecraft and operated for over 10 years. And, you know, MODIS came and other things, and other countries work on it. And I would just want to show you, oops, I don't know, it doesn't work. Maybe it is. I just want to show you orbits, one-day orbits of Seastar. So it covers most of the world's oceans with some empty space here. So in a couple of days, you would cover the entire globe. So from CWIFS, you can have a lot more data and maps like this. This is based on 10 years. You can also produce, of course, other data products like POC, particulate organic carbon using similar approaches, empirical approaches, or particulate inorganic carbon. This is from MODIS Aqua. Well, the bottom line is that using this initial, observations that chlorophyll and optical property correlates, this provided impetus for building empirical algorithms for chlorophyll, later on for other things. And this is how it all started. And today we of course work on many other approaches and semi-analytical. There are a lot of them around, and still, work is going on in this direction. But important thing is that, it's important to work on other approaches, not just empirical, because these provide a good trend over big range, but there's still significant natural variability. And that's why we are trying other things as well. So I would like to spend the last 7-8 minutes on one potential avenue of research, maybe some of you may consider to do, where we actually look at water as a medium consisting of many types of constituents, each of which has distinct optical properties, but at the same time may play a specific distinct role in ocean biogeochemistry. So that if we're able to define categories of particles, I will focus on particles, not dissolved materials, in some meaningful way, perhaps optics in the future can play even a bigger role than it does today for understanding ocean biology and biogeochemistry. So the idea is, and Zhangping touched upon this thing, that you could represent the bulk optical property of particles assemblage as a sum of different groups of plankton, possibly minerals, maybe even detritus. Some of this is maybe a dream today, but it doesn't mean we shouldn't think about it. And I will show you one example model that if you have information about optical properties of individual particles, you can, I will show you the forward models, how you do it. And this is based on the database that I showed before, 18 species of plankton. So, okay, so let's say we know optical cross-section, absorption, scattering cross-section, and things like that for each and every group. We can mix them at will at different proportions, but something that may be reasonable for seawater, so different numbers. We also know how much chlorophyll typically per cell is for these groups, so we can also keep track of how much chlorophyll is within group, and we can keep track of the total plankton and total chlorophyll. So this particular water body that is modeled with this mixture has 0.7 milligram per cubic meter, has this kind of distribution of numbers, but this water also contains detritus, minerals, and some bubbles. So the water looks like this. Each of these green lines is specific population of plankton. Well, this is supposed to simulate the minerals and organic detritus, and this is supposed to simulate bubbles. And so if you have a mixture like that, you can compute the total absorption coefficient, total scattering coefficient, and you have you have all plankton, minerals, organic detritus, and then you can further partition this curve into all plankton components, 18 plankton components. So I'm showing this just to give you an idea how powerful something like this may be in developing understanding how different things contribute. Whether or not this particular simulation represents any specific water body is another question, but it is a reasonable set of assumptions about these concentrations of these different things. You can also move a step further and use these IOPs, different IOPs associated with different components, which are characterized by number, concentration, and cross-section associated with these components, as input to radiative transfer model. And this exercise actually Kurt Mobley and I did late 70s, what am I talking about, 90s.
Yeah, early
90s. So we used hydrolite and data on different species. So we were feeding hydrolite with different scenarios, and you could really see what happens, for example, in terms of reflectance or any other things or light fields. So you can look in questions like what ways does variability in detailed seawater composition determine variability in reflectance, or what information about water constituents and optical properties can we hope to extract from remotely sensed reflectance. And this is just one example or a couple of examples from the simulations with hydrolite, when you see this reductionist approach is being combined on IOPs, is being combined with radiative transfer to simulate remote sensing reflectance. You can get very interesting results. You can, for example, okay, this is the one ocean that consists of water molecules only, so pure water reflectance. What happened? We can ask questions. What would have happened if the ocean contained only viruses in it, nothing else? The reflectance would look like this. Would be someone enhanced here, and all other kind of scenarios are here. So these are bacteria, the chlorophytes about 8 micron in size, cyanobacteria, diatoms about 4 micron in size. You can see pretty strong effects if we add one component, one specific group of plankton into water, or we have everything here as well. But another interesting simulation and scenario is shown here. In this scenarios, we used only this. We didn't use 18. Okay, in this example, it's not that many. But still, we are making our point. So here are two simulations. We have two water bodies in the simulation with 1 milligram per cubic meter of chlorophyll. And, but in one situation, we have chlorophyte bloom. These are the species that are about 8 micron in size. In another case, we have bloom of cyanobacteria. We call it a bloom. It's kind of 1 milligram. Is these guys, 1 micron and 8 micron. So two water bodies, the same bulk concentration of chlorophyll, but somewhat different composition of plankton. And you see very different spectra of reflectance. So if you apply blue to green band ratio algorithm for these two cases, you would derive two different chlorophyll concentrations. And yet this is showing that we have just one. The water body is the same in terms of chlorophyll concentration, but different in terms of plankton composition. Julia will talk next week about plankton diversity, deriving plankton diversity from optics. So maybe that relates a little bit to it. Okay, I have several other slides which I will skip because this is about the same point about the utility or how powerful the use of detailed description of seawater composition is in understanding the bulk properties of the ocean, bulk optical properties, including ocean reflectance. And I would like to close my lecture with this statement here that the complexity of seawater as an optical medium should not deter us from pursuing the proper course in future research. Perhaps some encouragement to you to embark on some challenging avenues in your research. And Steven Weinberg, who received Nobel Prize in Physics in 1979 for his work on elementary particles in physics, he said that the reductionist worldview has to be accepted as it is, not because we like it, but because that is the way the world works. So thanks for your attention.